Good day to all. In today's video, let's discuss about antigen and antibody interaction. So the antigens and the antibodies combine specifically with each other, and this interaction between them is called antigen-antibody reaction or antigen-antibody interaction. So these form the basis for humoral immunity or antibody-mediated immunity, and this reaction forms the basis for the detection of infectious disease-causing agents and some non-specific antigens like enzymes. When antigen-antibody reaction occurs in vitro, they are known as serological reaction. So the reaction between the antigen and antibody occurs in three stages as follow. So in the first stage, the reaction involves the formation of an antigen-antibody complex, and in second stage it leads to the visible events like precipitation or agglutination and etc. So here we can able to see the precipitation or a clumping of antigen antibody visibly. So the third stage includes the destruction of antigen or its neutralization. The salient features of antigen antibody reaction are as follow. First is specificity of antigen reaction. So the specificity refers to the ability of an individual antibody combining site top react with only on antigenic determination of the ability of a population of antibody molecule to react with only one antigen. The antigen, it will be very specific to bind with a specific antibody for forming an antigen antibody reaction. And next is immune complex. An immune complex is formed from the integral binding of an antibody to a soluble antigen, and the bound antigen acting as a specific epitope bound to an antibody is referred to as a singular immune complex. And the antigen antibody complex formation results in complement activation and opsonization of target cells. Binding site of antigen antibody reaction. In the antigen antibody reaction, the antibody attaches with the antigen, and the part of antigen which combines with antibody is called epitopes. And epitopes also known as antigenic determination, and it is the part of antigen that is recognized by the immune system, specifically by antibodies, B cells or T cells. So the part of antibody that recognize the epitopes is called paratope. And binding force of antigen antibody reaction, the binding between antigen and antibody is in antigen antibody complex or reaction is due to three factors, namely closeness between antigen and antibody, and non-covalent bond, and affinity of antibody. So closeness between antigen and antibody is closely fit, and the strength of the binding is great. Covalent bonds, the bonds that hold the antigen to the antibody combining site, or all non-covalent in nature. So this includes like hydrogen bonds, electrostatic bonds, van der Waals forces, and hydrophobic bonds. And affinity of antibody. Antibody affinity is the strength of the reaction between a single antigenic determination and a single combining site on the antibody. Avidity of antibody. So avidity is the strength of the bond after the formation of antigen antibody complex. So it is used to denote the overall capacity of antibodies to combine with the multivalent antigen. A multivalent antigen has many types of antigenic determinants. And cross reaction, an antiserum raised against an antigen can also react with a similar antigen of another type. A similar type of antigen can also form an antigen antibody reaction, which is called as cross reaction. And this is called as cross reaction, and the antigen which produces the cross reaction is called as cross reactive antigen. Thank you.